A muscle fiber is packed with microfibrils. 80% of fiber volume consists of sarcomeres. What sarcomeres are, as you can see here, what's circled in the red color, are lines arranged in parallel and in series, and they are the smallest functional units of muscle. You have here the A band from the start of the dark band to the end of it, and you have the light band is called I band. This is where the concentration of the proteins actin and myosin are. The darker the band is, will have more concentration, or the thicker protein, which is the myosin, and the lighter one will have the thinner concentration, which is the actin. The M line is also called the midline, which separates each Z discs from each other. The Z disc indicates the beginning of each sarcomere, which means the space between each Z disc, this one and this one, is called a sarcomere. You can see here the structure of the myofibril. Consists of the Z disc here and here. The space between two Z discs, as you can see here, is called a sarcomere. The thin filaments is the protein actin, and the thick filaments are the protein myosin. The space between two thick between two thick filaments, or between two concentration of myosin, we're going to call it an I band. Okay? And the space between them is called an A band. This, of course, is the age zone or the midline. As you can see here, it says each sarcomere extends from one Z disc to the next. The myofibril are bundled enclosing from 100 to 400 thick myosin filaments, about 1 micrometer in diameter, and they make up 80% of the volume of muscle fiber. The adult muscle contains about 2,000 myofibril. The myofibril is a part of a muscle fiber, okay? So the myofibril is the smallest part. We're going to take a look at the sarcomere. As we mentioned before, the sarcomere is the space between two Z discs, okay? The red ones are the myosin, which is the thicker filament. The blue ones is the actin, which is the thinner filament. The yellow ones is a protein that will have contract and relax, sometimes called a filling or a titin. In this diagram, we have here the fully contracted sarcomere of a muscle fiber. In the contracted state of the sarcomere, you can see how the thicker are the myosin, as we said here. The thicker filaments are the myosin are stretched, and the thinner filaments, which is the actin, is also stretched. Since the actin sort of similar in shape as a spring. So in the contracted form, you will see its full, extended, coiled upon itself form. And the titans are the fillings, and the yellow structure is also extremely uh, tight and coiled on each other, on itself. This is the M line, the midline, and this is the age zone that separates between two thick filaments. The space between Z line and Z line is called a sarcomere. So if we want to represent it by a more simple diagram, you have the orange part as the midline. The green ones, or the space from the red ones, are the eye band, and the entire bit is called a sarcomere. In here, we can see the enlargement of one sarcomere section lengthwise. This is not a cross section. Uh, this is a length-wide section. Notice the myosin heads on the thick filaments. This is the enlargement of one sarcomere, and this is in the contracted state, and this is in the enlarged state. The blue ones are the thin actin filament. The yellow ones are elastic filaments, as we said before, they're titans. And the the red ones are the thick myosin filaments. The sarcomere consists of cytoskeletal protein, such as filamentous actin and the motor protein myosin. We have here the sliding filament model. In the first one, we're going to notice the difference between the state of a fully relaxed sarcomere against the fully contracted sarcomere, and the state of the muscle which contract and relax. As we've said before, the muscle will of the tricep and bicep will work in the antagonistic state. So we're going to look at what happens when, let's say, when the bicep contracts and the tricep relaxes, and vice versa. Since each of these muscles are obviously made from filaments and myofibrils, so we're going to look at the state to notice the difference of them. First, notice the Z line or the Z disc. We have the space between them is called the H line. And this area is called the A line. Let's look at this in the relaxed state. First, let's look at the thick filaments containing the myosin. You can see it in its normal state in the filament. There aren't any changes or any pressure applied to the filament. Now, let's go to the actin. The actin is also in its relaxed state. There are no significant changes to the fibers of actin. Let's look at the yellow ones, which is the titan or called elastic filaments. You can see the coiling of the elastic filaments or the titans in here is normal, like a normal spring. They are also kind of uncoiled, as you can see here. Because of this relaxing state, and there are no pressure or force applied to it, so we can see it in here being a normal state with no pressure applied, and then they are also kind of uncoiled. Now, let's look at the fully contracted sarcomere of a muscle fiber. Before that, you can notice the space of, or the proportional space of the Z line to the A line. Okay. In the case of the fully contracted sarcomere of a muscle fiber, you can see how the actin is attached to the midline. In the relaxed state of the myosin, the actin are not attached to each other. They're sitting in their own place. However, in the contracted state of a sarcomere, you can see how the actin fibers are got closer to each other and got attached to the midline. In the case of mycin or the thick filaments, you can see it being stretched. The stretching of the myosin fibers is affected by the titin. You can see how it's being fully coiled. The coiling of this titin will affect the mycin by pulling it to the side. Once it's pulled to the sides and it became in the state of contraction, okay, it becomes more coiled, as you see here. You can look at the difference between the two diagrams, the coiling of it. This becomes extremely coiled upon itself. This is uncoiled and it's normal state. We can also notice the difference in the space of the Z line compared to the A line. You can see how it got smaller. It got smaller because it contracted to itself. Okay, so let's assume the filaments of the model A is in the tricep, and the filaments of the model B is in the bicep, since they work in an opposite way of direction. The first one is relaxed. You can notice from the uncoiling of the titans and how the actin fibers aren't attached to the midline in any shape or form. In the relaxed, in the contracted state of the muscle fiber, you can notice how the um, titan fibers are coiled. Their coiling will drag the mycin fibers outwards, which will stretch them even more, and the uh, actin fibers are attached to the midline. In this picture, you can see the fully relaxed state of muscle fiber. You have the Z line, you have the A line, and you have the other Z line. You can see the state of the myosin fibers and the actin fibers. Also, you can see the titans. Notice the difference between the fully contracted sarcomere and the fully relaxed sarcomere. In the second picture, everything is being stretched, okay, due to the action of contraction. The, the titans are being coiled, the myosin is also being stretched, and the actin touching the midline. You can see the fine white line over here. This is the space of where the actin leaving space and not attaching to the midline. However, in the contracted state, you can see that it stretches and attaches itself to the midline. We have now the actin myosin bridge, cross bridge cycle. The interaction of actin and myosin to produce force. So the cycle is done to produce force. ATP, which is the adenosine triphosphate, is needed to separate the primes of to separate and prime the myosin heads, which give it a stored elastic energy. Myosin freely binds to actin, releases stored energy, and contracts, moving actin and Z line. Let's look at this cycle, the cross bridge cycle of actin and myosin. In the first picture, you can see the cross bridge formation. You can see here the myosin, which is the thick filament in the red, and the blue one is the thin filament. This structure that is going out from the myosin is called a myosin head. We have an ATP molecule, and you have a calcium, and you have a phosphate molecule, you have a calcium molecule. The first action of this cycle, of course, this is in the relaxed area since it's uncoiled and the actin isn't attached to the midline. After knowing each of the elements in this picture, we're gonna see what first happens. The calcium switches the machinery on. The myosin heads bind to actin. When this process starts, the calcium molecule, this one, will start an action. After starting the action, this structure, which is the myosin head, will rise and attaches itself to the actin filament or the thin filament. In the second picture, you can see the ADP molecule, which is the adenosine diphosphate molecule, and one phosphate molecule is being released. And the structure of the myosin head is moving to the left with the moving of the actin, with the moving of the thin filaments. This is in the case of making energy for contraction because we're starting in a relaxed state. Of course, the ATP and molecule and the phosphate molecule is the stored energy we talked about. You can see it sitting on the myosin head. Once the machinery starts, the myosin head starts moving and the thin filaments will contract and moves to attach itself to the midline. In this case, the energy, which is the ATP molecule, is released. Okay? Once the energy is released, the myosin head moves. And the state of the ATP molecule and the phosphate doesn't give us energy. So, in a chemical formation, the phosphate molecule will be added to the ATP molecule and it will turn into an ATP, which is an adenosine triphosphate. Once the ATP attaches itself to the myosin head, the cross bridge attachment, the cross bridge formation. So, the ATP will attach to the myosin as an elastic energy. As you can see here, by this motion, it means the myosin head will bounce up and down, attaching and detaching from the thin filament. The chemical energy in the ATP is transferred to myosin as elastic energy. Once the ATP is sitting on the head of the myosin, it will break down to its original form, which is ADP molecule and a phosphate molecule. This process is called the ATP hydrolysis. And once it comes back to its original form after undergoing ATP hydrolysis, we will go back to step one of the cycle.